My name's Ria, and I'd like to share the information I learned from how human gene therapy has treated brain cancer since 1990. Who I am Ria Sharma. I live in Pleasanton, California. I love books, fencing, and playing the piano. I am in 8th grade, and my favorite subjects are biology and engineering. A combination of the two fields is bioengineering, and a subcategory of the field is genetic engineering, which I find very interesting. Since I'm interested in doing research to help humans, I decided to explore gene therapy. Since I've always had an interest in cancer, I decided to research brain cancer. Objectives. First, we will cover the history of genetic engineering and brain cancer. Then we will move on to talk about the current discoveries on human gene therapy treatments for brain cancer. And lastly, we'll talk about some futuristic techniques that can be used. Background information. Definitions. There's brain tumor, which is a solid mass of tissue that forms when abnormal cells group together in the brain. There's benign, which is not harmful in effect. There's malignant, which means very virulent or infectious. There's the glial tissue, which provides support for an essential nervous system function. There's the glioma, a malignant tumor of the glial tissue of the nervous system. There's the glioblastoma, a highly invasive glioma in the brain. There's IDH or isocitrate dehydrogenase, which converts isocitrate to alpha ketoglutarate, pivotal in cellular metabolism. So there are two types of brain cancer: metastatic brain cancer or secondary brain tumors, which is cancer that originates in another part of the body and spreads to the brain. There's primary malignant brain tumors, which is cancer that originates in the brain and spreads to other parts of the body. And there's the 5-year survival rate, which includes the people who will be alive 5 years after diagnosis. It does not include those who die from other diseases. The survival rate is calculated based on the following factors: the type of cancer, the stage of cancer, the age at time of diagnosis, and the time period. A fact you'd like you to know is that approximately 90,000 people are diagnosed with brain tumors in the US yearly. So brain cancer is a life-threatening disease due to changes caused in the vital structures of the brain. In 2020, an estimated 251,329 people have died worldwide, both of primary brain cancer and the central nervous system or CNS tumors. There are two types of brain tumors: malignant brain tumors or brain cancer, and benign brain tumors, which are tumors who do not spread and are removable through surgery. So here's a table that shows you the 5-year relative survival rate for the certain age groups of malignant brain tumors. For the pediatric patients or ages 0 to 15, there's a 75% chance. For ages 15 to 39, there's a 72% chance. For 40 plus, there is a 21% chance. So treatment methods before gene therapy. There's radiation oncology, which is medical treatment that uses radiation to kill cancer cells. There's surgery, which has been going on from 1886 to present day, which is a procedure to remove or repair a body part and is used to find out if a disease is present in the body. There's drug treatment that uses powerful chemicals to kill fast-growing cells. So gene therapy and genetic engineering are useful since it gives long-lasting effects with the potential of eradicating disease. This makes sure that side effects are a rare case. Genetic engineering. Genetic engineering is the artificial modification, manipulation, and assimilation of DNA in order to modify an organism. It's usually in reference to recombinant DNA technology. Here is a timeline that shows you the different advancements in genetic engineering. For example, in 1985, we have the human growth hormone. In 1986, we have hepatitis B vaccine. In 1986, also, we have the first disease-resistant plant. And in 1982, we have human insulin. So key findings across papers. So there are many diverse approaches and techniques for human gene therapy. Some diverse approaches are gene transfer-based immunotherapy, gene-directed enzyme project therapy or GDEP, and oncolytic virus therapy. Some techniques are calcium phosphate transfection, which involves DNA and calcium phosphate added to cultured cells. There's electroporation, which utilizes electromagnetic waves to make cell membranes permeable to DNA, enabling gene expression. And there's plasmid utilization. Plasmids are circular DNA molecules injected into tissue. So here's a table comparing the different methods of gene therapy. I like to point out the integration, which is if you see these different viruses in retroviral, it does integrate. In adenovirus, it does not. In adeno-associated virus, it does. In herpes simplex virus, it does not. And in naked or lipid DNA, it's very poor. And for the duration of expression in vivo, we have retroviral, it's long. In adenovirus, it's short. In adeno-associated virus, it's long. And herpes simplex virus is short, as well as naked or lipid DNA, which is short. So that's why we can see that retroviral and adeno-associated virus is the ones used the most. So the viral methodology of human gene therapy. The viruses are modified for delivering therapeutic genes that are such as adenovirus, herpes simplex virus, adeno-associated virus, and retroviruses. The selection of viral vectors are crucial because it impacts the therapy effectiveness with advantages such as tissue selectivity, integration into the host genome, and stability. There are some drawbacks like tissue toxicity, immune reactions, and limited gene size. There are delivery challenges that exist for viruses penetrating the brain. So there are ongoing efforts to enhance delivery via intra-arterial injection with blood-brain barrier disruption. Overall, gene therapy using viral vectors shows promising results in reducing brain tumor size and growth. Here, this table shows the 5-year relative survival rate for those with brain cancer or CNS. So for the next paper, here are the introduction goals and outcomes. Here are two images that show baculoviruses and adenoviral vectors. So the study evaluates baculoviruses or BVs as gene therapy vectors for brain cancer treatment, using cell cultures, animal models, and human gene therapy. Researchers aim to understand BV's efficacy as vectors for gene therapy, particularly their immune response and capacity for gene delivery into the CNS. Adenoviral vectors, or ADVs, and baculoviral vectors, BVs, are used to transduce normal and tumoral glial cells. ADVs showed higher efficiency in transferring genetic material compared to BVs, with over 90% transduction efficiency for ADVs versus over 40% for BVs. And IDH focus and outcome. Genetic and epigenetic alterations such as mutated IDH SAS are crucial in classifying diffuse gliomas and determining prognosis. BVs and ADVs are utilized to transduce neurospheres derived from glioblastoma patients, with implications for gene therapy in mutated IDH gliomas. Both viruses show linear dose-dependent enhancement in transduction efficiency without inducing cytotoxic effects in astrocytes or neurospheres. A continuation of an IDH focus and outcomes. ADVs exhibit higher transduction efficiency in neoplastic astrocytes, while BVs demonstrate higher efficiency in normal astrocytes. In mice, the highest BV dosage tested did not induce neurotoxic effects, while the maximum tolerated ADV dosage was determined for intracranial injection. BVs demonstrate stability in transgene expression for at least 21 days without neurotoxicity, highlighting their potential for gene therapy in brain tumors. No, it is not easy to deliver viruses to the brain. So the key point of the paper was that the article focuses on the use of baculoviral vectors and different techniques in animal models to effectively treat brain cancer. Lastly, a futuristic aspect. So in the future, there's a potential to eradicate various cancers by leveraging the techniques on this research findings. So in 1985, we had the human growth hormone, then we had hepatitis B, then later on we had CRISPR-Cas9, which was invented in 1987. In 1990, the first federally approved gene therapy treatment is performed successfully on a 4-year-old girl. In 2006, we had the FDA approve a recombinant vaccine against human papillomavirus, and in 2016, we have CRISPR-Cas9 first used on humans. So there are upcoming advancements still going on in gene therapy. Hopefully, one of them soon is in cancer. Acknowledgements. I'd like to thank my mentor, my parents, and my showcasing specialist. Here are some of my references. Thanks. If you have any questions, feel free to email me.